Right, so thank you very much for inviting me. I feel quite self-conscious following both William and David, who are both very established, and while I'm still very much in the process of trying to establish myself. So I guess I want to tell you a little bit about where I've come from and what it is that I do now. So first of all, my name's Lena Ciric. I'm now a lecturer in environmental engineering at University College London in the Civil, Environmental and Geomatic Engineering department. But I started off working at CEH. Well, I did my PhD at CEH looking at the communities in diesel degrading consortia in groundwater. I then moved into looking at the communities in the human mouth and trying to develop mouthwashes, anti-gingivitis mouthwashes from functional foods. So our best active one was made of shiitake mushrooms. Didn't taste very nice. However, I then went on to work on another postdoc where we were looking at the effects of antibiotics on the various microbiomes around the body, and I was specifically looking for new resistance genes and elements. And then at that point I got lucky and got a lectureship, moved into another department. So I was at the Eastman, sorry, for my two postdocs I was at the Eastman, which is also at UCL, and then I moved departments to civil engineering. So now my job is to apply all of my microbiology knowledge to thinking about how infections move within the built environment. And by that I mean places like this. So, well, I guess it's sort of like a classroom. So teaching spaces, schools, obviously hospitals. Hospitals are the obvious choice, but there are a lot of people working in hospitals, so I'm trying to move away from that as much as possible. So offices, schools, but also transport systems, which I'm very excited about, but not getting very far with. So I'm going to give you a bit of an overview of the stuff that we do. I'm pretty much going to give you, show you just two examples because it's still early days for me, and it's just to get you thinking, because I think I come from the same sort of background as most of you, whereas where I work now is very different. So it's actually been very interesting to work with engineers and see how they don't understand where I'm coming from most of the time, and how they're obsessed with trying to apply things, which I think is good, and it works with me. So that our logo for UCL Engineering, my faculty, has changed the world. So you know we're pretty confident and perhaps overconfident, let's say.
Ours at Southampton has changed the world for the better.
Yes, yes, exactly. Yeah. So. So my department is engineering a better world. The whole of UCL is just changing the world. Okay, so I now run the Healthy Infrastructure Research Group. So the main things that we do are investigating the pathways of the spread of infection within the built environment, and then also developing and testing engineering solutions to reduce the spread of infections. I don't think we'll be able to eliminate it altogether. So I guess this diagram just shows you the sort of the sort of interactions that we're interested in seeing. Blessing everyone who's having a sneezing fit. Oh, no, sorry. So we have, so this is within the civil engineering department, so we have for them a very exotic grade category 2 microbiology lab. For you, not exotic at all. But for you, we do have an exotic environmental chamber, which is a generic room size. So I think it's 4 by 4 meters, 2.5 meters high. And in there, we can aerosolize various non-harmful bugs and see what happens, see how they settle, where they settle, how long they'll be viable for, and so on. And we also have this very nice mannequin in different fabrics that we were trying to see whether things survive for longer on different cloth, but we couldn't get any bugs out, so that was that. Anyway, they're there to scare us every time we go in. And we have this smaller chamber, which is within the bigger environmental chamber where we can run smaller experiments. So I should say that the environmental chamber, we can change the temperature, humidity, and pressure in there. So, and we also have a control over the air mixing in there and also the air changes. So it sort of mimics being in an air-conditioned room where the air isn't being changed much, or being in a non-air conditioned room where you're changing the air. So anyway, I will explain that better when I move on to the next example. So this is my first example. It's a very simple piece of work, but I think it's quite interesting, and it will give you a good idea of the sort of thing that we're trying to do. So this project, we were approached by Public Health England while they were still the HPA, and they were interested to know, so this was the TB specialists in particular, and they were interested to know whether it's possible to quantitatively measure an environment that would give a high risk of TB transmission. So what happens is that every time somebody goes to their GP with TB, the HPA is flagged up, the PHE is flagged up, and they have to go and do contact screening if this person has been in a workplace or in a school, and see how far the infection might have spread. So normally they would go into a place and say, this is cramped, not very well ventilated, it seems like there might be a high risk from the environmental factors, but they wanted to put a numerical value on that, and this is where we came in. So what we did was come up with a set of measurements that we could do in a room like this. So we'd look at what the ventilation was like, whether the air was being mixed by air conditioners, how many people there were per, or how many square meters floor space were there per person, and all sorts of other things, what the ventilation system was like, and then see whether it was in any way correlated with the number of people that got infected. So I must confess that I'm neither I'm not a TB expert, so please bear with me. So this was the first case study that was done. This was an office, so this is our office here with the various desks, our index cases here, and then you can see all the latent cases marked with L. So the air conditioning units are marked as these little boxes, and then we've got the ventilation outlets in the toilets, but not much in the way of ventilation inlets. So this graph here on the right is showing you the carbon dioxide measurements through 3 days, and these our devices were measuring at points A and B, so these are the ones closest to the index case. And you can see that the carbon dioxide rises over the day, which is what you would expect. But we can also, so the carbon dioxide rises over the day, so this to us suggests that the ventilation within this room isn't very good. So the people in here were all very happy because it was nice and cool, a bit like it is in here. So the air conditioning was working, and it seemed like a good environment. However, all the air conditioning was doing was spreading the stale air around and pumping it round to everyone else. So we had so it wasn't just the people that were close to the index case that were getting infected. There were some as far away as they could be as well. So our conclusion from our first case study that we did was that in this case, poor ventilation and well-mixed air resulted in basically the airborne infection spreading around the office. And the Wells-Riding model was applied here, and we found that our model predicted about a 42% infection rate, and the infection rate was 50%, which is very high. So PHE pretty much say that if the infection rate is over 10%, then that's high. So we've then gone on to work with them further. So the team, so this is the London TB Extended Contact Screening Team. So they've been funded for a few years. Funding's due to run out soon, so I guess that'll probably be the end of that. But they've been looking at various infection measures, so I'll go through those in a second, and then we've been looking at the site itself. So then what we wanted to do was take their infectiousness data, our environmental data, put it together, see what correlates most with see what environmental factors correlate the most with the infection rates, and then develop a risk assessment methodology. So I guess I just wanted to underline that it is very simplistic, and I completely agree with that, but what we have been able to do is put a quantitative value on the environmental risk. And although this was specifically for TB, I think it's something that could be applied to all sorts of other infections, and also, well, airborne infections, and also something that could be used as a guideline for facilities managers to make sure that spaces are healthy for us to be in. And I think this is something that, I guess it's certainly a luxury of the developed world to be able to worry about things like this, you know, making sure everything is nice and we're all very comfortable and happy everywhere. But I don't know, I think it's good. I think it's good to strive for something good. So this is the other example I wanted to show you. So we have been working with some colleagues at the UCL Department of Chemistry, and they've developed a light-activated antimicrobial surface. Well, it's a concoction that they've been able to incorporate into silicon, and we've used that as the surface. So we've tried a range of different microorganisms. We basically just put viable organisms on the surface, left them to incubate for a while under white light, and then took samples at time intervals to see when they were inactivated. So I'm going to show you the results for the different microorganisms, but it's only Staphylococcus epidermidis that we looked at with transmission electron microscopy. So again, I'm not an expert in chemistry, but this is how it works. Bear with me. So the photon hits the photosensitizer, which is this molecule here. The photosensitizer goes from its ground state to a singlet state, which releases an electron, which then reacts with substrates. So that could be, in this case, various microbial components like proteins, lipids, nucleic acids. On the way back round, the singlet state moves down to the triplet state, and this triplet state actually interacts with molecular oxygen in the air, and that releases singlet oxygen, which then reacts similarly. Whoops, sorry. Oh, this suddenly started working. Then reacts like the electron with various substrates. So again, various parts of the microbe. So the hope is that because the system works with all sorts of components of the microbe, it would be difficult for it to become resistant. But it only does work in light, so that is a problem. So this is how the chemists make the surfaces. They basically dip. So this is a mixture of methylene blue, crystal violet, and nano gold particles. They basically swell the silicon, which allows it to take up the gold nanoparticles and the methylene blue, then air dry it for 24 hours and wash and towel dry, and then dip it in crystal violet. And they've been able to use this to impregnate lots of different materials, actually. But we've been working with just the silicon. So the combination of the methylene blue and the crystal violet means that the activation, it gives us a wider spectrum of activation, so a wider spectrum of light can activate the surface and have the antimicrobial effect. Yes, I just want to conclude that the surfaces were effective, and that from my point of view, thinking about the built environment, I think it would be good to incorporate them on high-touch surfaces in public spaces. So, well, in hospitals, definitely, but possibly maybe, I don't know, grab poles on buses or something like that. The other thing that I've been doing is schmoozing London Underground, and they are interested to know which parts of their stations and trains are the most contaminated so they can clean them better. What I want to know is exactly what's there and how resistant it is to everything. So we have different interests but the same sort of aim. So watch this space. Another project I'm involved with is using light-activated nanoparticles to treat water. And then I have a student coming in September who's going to look at antibiotics and resistance to them in various parts of drinking water treatment. So yes, looking forward to that. And I think I just want to thank the team at UCL in my department, as well as the chemistry team, partners at NHS from Elaine Clampman-Green from Great Ormond Street and Peter Wilson from UCLH, and our collaborators at Public Health England. Thank you very much.